The overall goal of this technique is to extract bacterial periplasmic contents by cell fractionation for biophysical characterization, including X-ray fluorescence, X-ray crystallography, and radiometal uptake.
This method can help us understand key biological relationships, including structure-function relationships of ABC transport systems and how they recognize their substrates specifically and transport them into the cell through the lipid bilayer. The main advantage of this technique is to separately evaluate the cell contents and to extract protein from its native environment with minimal cross-contamination.
We first had the idea for this method while trying to remove YFEA substrate, realizing instead of using an artificial approach, we could use the cell to produce apoprotein.
In addition to providing insight into substrate binding and transport across bacterial cell compartments, methods like this radiotracer assay apply to eukaryotic systems, which also have robust fractionation techniques.
First, thaw a previously prepared bacterial subculture resuspension at 4 degrees Celsius. Then, pellet the thawed cells at 4,000 times gravity for 20 minutes at 4 degrees Celsius. Next, resuspend the cells in 50 milliliters of ice-cold high-salt buffer by aspirating with an automated pipette and a 25 milliliter tip. Incubate the suspension over ice for 20 minutes, with occasional inversion for mixing. After incubation, pellet the cells at 4,500 times gravity for 20 minutes at 4 degrees Celsius. Resuspend the cells in 50 milliliters of ice-cold low-salt buffer by aspirating.